All right, we're going. And so you can draw a line from the carbonyl to the nitrogen, and if they're those arrows or draw an arrow, if those arrows are going different directions, it is anti-parallel. So this one is anti-parallel. Okay, and if you're going they're going the same direction, then it's parallel. Questions on that? Okay. So then let's go on and talk about turns. Here we have type 1 turns, we have type 2 turns. I'm sure there are more names for various turns. These are called beta turns. We're just going to call them turns and classify them all in the same category. In the case of these turns, we have 4 amino acids. I've seen them with 3 amino acids, but there are some characteristics that are common to all turns. Excuse me. So we talk about turn characteristics. What forces are holding these turns in place? What's the primary force? Hydrogen bonds, right. You see that hydrogen bond right here? So you got a hydrogen bond between the carbonyl of this amide and the nitrogen of this amide, the hydrogen on the nitrogen of this amide. And it's all of our hydrogen bonding has been between the carbonyl of the amide of one amino acid and the nitrogen on the amide of the other amino acid, okay, for all of our secondary structures. That's what's going on. So hydrogen bonds hold the shape. All right, and so we've got the hydrogen bonds holding us in that shape there. Previously, we've had amino acids that we kind of didn't like to have in these secondary structures. Really, for beta sheets, we didn't care so much, but certainly for alpha helices, we had amino acids we didn't work real well. What were those?
Proline and glycine.
Okay, what does proline do?
It's too rigid.
It's too rigid, and it sort of forces a turn, right? So actually, here what we have is our glycine and our proline no longer are the black sheep of the family in terms of amino acids. They're actually good in turns. We'll see them more in turns. So proline is more common. And when I say more common than normally you see it, because it forces a turn. So we'll say its connection to the backbone. So there's a side chain, let's say its side chain connection to the backbone. Forces a turn. And glycine's more common because it's super flexible, so it accommodates the turn better. Okay, so that's turns. We're not going to say a whole lot more about them. I did want to give you an example. I want to do this in PyMol, but PyMol apparently hates me today. So if we look here, I'm going to get rid of our waters. I don't know if we have ions. Nope, no ions here. But this is hemoglobin. So this is human hemoglobin right here. And you can see what secondary structure makes up most of hemoglobin. Yeah, it's mostly alpha helices, okay? And so these alpha helices are running in a straight line, and then they get to the edge of the hemoglobin, and what do they have to do? Well, if we look at this one right here, not all of them are, sometimes we've got a bunch of squiggly kind of unstructured, but it's probably structured because this is a crystal structure, but it's not one of our secondary structures. We call this random coil, although it's not always random. But here we have a sharp turn. I'm going to go ahead and turn it so you can see it better. So It's just going to be against everything else. You can see that we have this helix right, I lost it. Colors, I could have chosen something with better colors. I thought I just had, I mean, up here you've got something going off into space and it needs to turn around and come back into the protein. Okay, here. Here we got something moving along that has to go back into the protein. Okay, so the turns, you have to turn around, otherwise your alpha helix just keeps going on forever. Questions? That's an example of that. When I was looking this up, I found something interesting. So let's take a look at this as well. So Protein Data Bank does this fun stuff where they have like a molecule of the month over here. And so these things are called incretins. I'm not sure how to pronounce that. I've only read it. Okay, but that's our molecule of the month. What do you think the second, so that's actually, it's a protein. What do you think the secondary structure is there? Or the full, it's really the 3D structure. Well, we have names for, what name would you give to this structure? Yeah, it's an alpha helix. Do you remember the other day when I put up the alpha helix and I stripped off the side chains and what it looked like? It looks a lot like that, right? That actually has the side chains in it. So what does that tell you about the side chains or the amino acids that are in this? What was that? Well, they're going to be hydrophilic, right? Because this thing is some molecule that, well, it's out by itself, right? So it's probably not something that doesn't go into solution. So I'm going to guess they're probably fairly hydrophilic. Otherwise, it wouldn't dissolve. What else? What else does it tell you about these side chains? They do stick out, and it still sort of looks like an alpha helix, right? So they can't be that big. It's got to mostly have kind of small side chain amino acids on it. Okay. And if you're ever interested in looking at these things, so they have a nice little thing, they talk about it. So these are hormones that are released after you eat a meal. I did not have time to look at this, but I see insulin, and I'm wondering if these are related. I think I've heard of GLP-1 targeting drugs. Let's see. Screening hormones dependent, blah, blah, and glucagon-like peptide, GLP-1s. I think this is related to Ozempic and those other drugs. Okay. I think that they might be like this, but sorry, I didn't have time to read it ahead of class. But you know, they do bind to a receptor, right? So bind to a receptor that will signal certain things. It's probably signaling satiety in some way. Anyway, mostly I was interested in, hey, that looks like an alpha. I looked at that and went, oh, it looks like an alpha helix, right? And so actually we can see that if we, hold on. We can't see that in any of their pictures they show us, but what we can do is we can use the protein data bank. We can go and we can look up these structures, right, and see them in more detail the way we want. So you can go and find these four-letter code structures. I guess I can probably even click on this. Look at that, it was even easier. And I can do that, go to my structure, and there it is. You see that it's in, this is it right here, right? You see that's like from previous pictures, this must be it. And it is indeed an alpha helix. So lots of cool stuff, lots of cool tools out there you can use. But this is just we're just talking about proteins, looking at proteins. Okay, I want you to be used to doing that sort of thing. All right, then let's talk about one other structure. And I want to talk. This disulfide bonds is what I want to talk about. So it's something important. I'm going to call it a structure, but it's really just two cysteine residues that interact with one another. So these form when two cysteine residues link. And they're going to form a covalent bond when they do that. All right, this is a structure that will hold proteins in their shapes. They're not as important as hydrogen bonding and van der Waals, which are London forces, right? Get a whole bunch of London forces on the inside of a protein. Those hydrogen bonding and the London forces are more important to protein structure than disulfide bonds, but some proteins use them and need them. You can also have subunits of proteins come together. So for antibodies, you'll see that. Maybe we'll try looking up an antibody real quick. So these form, and so responsible for some protein 3D structures, or let's say contribute to. So I think maybe I won't try and find the antibody thing right now, but one example of this, well actually first off, let me draw a structure here. So you could imagine if I have a protein sequence running down this way, okay, so that's the backbone right there, and then off the one side chain might be a cysteine, right? And then if I have another protein sequence over here, and especially if it's a small protein, it's going to be hard to get those things to link together very tightly because it's not going to be able to make a lot of those London forces, right? You need to have a lot of London forces to make something very stable, but so we might have something like this. That's our cysteine beforehand. And then afterwards, we would have this. And now these two peptide chains or two parts of the same peptide would be brought together and linked together with a covalent bond. Is that an oxidation or reduction reaction? It's an oxidation, right, because we have fewer bonds to hydrogen afterwards, right? No oxygen involved, at least in the products, in the reactants and products. Questions on that? Okay, so disulfide bonds are important in a whole, think about keratin. When you think about keratin, what do you guys know about keratin? Yep, keratin is the protein that makes up hair and nails. It's also important in skin. Nails and horns. Yep, you see horns on some animal, that's keratin, okay? And when you think about it, and so we've got this same protein that makes up these different structures. Particularly, let's go with hair, nails, and horns. We get more disulfide bonds as we go from the left to the right, so it makes it more rigid. All right, so as we go from left to right here, in terms of whether you're looking at hair or nails or horns, what you're going to see is there's more, cysteine present in these. Okay, so you can get more rigidity out of that sort of thing. So let's think about that. If we go down here and we look at the structure of keratin, keratin is just by itself one protein unit. It's just an alpha helix. All right, so alpha helix goes on for a while. Apparently, we get some little knobs at the end. So you get some sort of little globular structure at the end based on this cartoon. That alpha helix, like most alpha helices, is which-handed?
Right.
Right. Okay. So if I look at this, I can do right. Now, if I take two of them and I put them together and wrap them around each other, let me move this down so I can make it larger for everybody. All right. So then we'll get two of these keratins that can come together and wrap around each other to make a coiled coil. Is that coiled coil left-handed or right-handed? It's left-handed. Okay. So if I put my thumb this way or this way, it works either way. It's curling around this way with the way my thumb's pointing. Okay, so we go and we make this left-handed coil, and then those coils can come together to make these filaments. And at this point, so the coil, what do you think's holding the coil together? Not hydrogen bonds. Not disulfide bonds. They're not disulfide bonds between the two there. I don't know. It depends on how I'd have to think about for the power of friendship how I feel about how I feel about that in terms of intermolecular forces. Give me another one.
The charges?
No, not the charge. Well, yeah, not the charges. Charge is a great guess. London forces, right? I'm gonna have a bunch of hydrophobic or sort of hydrophobic residues on going around one of these alpha helices. So if you followed it, you watch them go around the alpha helix in a way that two of these can come together and all those hydrophobic residues can come next to each other and hide from water when it forms that coil. Does that make sense?
So it's
Right?
hydrophobic interactions.
It's going to be a hydrophobic interaction, okay? Because that'll keep it away from water. If it was an amino acid that was hydrophilic, that could hydrogen bond, it could interact with another protein or it could interact with water, right? But if it's a hydrophobic one, it can't interact with water. These things are in water, right? At least when they form, and so you're going to have hydrophobic interactions that are going to be running along the insides of these coils, which I can't really readily demonstrate here, right? Does that make sense? It's going to be held together by those hydrophobic residues hiding from water on the insides, where the inside is where they're touching. Okay, and then you would have hydrophilics on the outsides. Okay, but now we have these protofilaments where we've got pieces that don't have any connection with each other. All right, and we could have hydrophobics on the outsides as well. I expect at some point the cells are going to have some problems making things, right? You can make something that's a cell can make something that's hydrophobic, but if there's too much hydrophobic, it's going to crash out of solution. So these balances that happen. All right, and so at this point, now we're going to get our disulfide bonds holding these protofilaments together. So I'm going to do this, and I'm going to say disulfide bond. So we get this protofilament, and that's going to give us this coil. That's what's going to hold these things together. Okay? And so this is what's in the structure of your hair, and it's also in the structure of your nails. And when you get if you get a perm, I've actually never had one. I did have hair once, so I could have, but I never got a perm. But they take advantage of this. When you get a perm, it smells really bad, right? Okay. Maybe it's gotten better, but when it used to be, at least when somebody did a perm, it would smell bad, and that's because they're using reagents to do the perm that are reducing and oxidizing. Okay, and a lot of reductants and oxidants smell bad. And so how does a perm work? Well, you'll have your hair that has those keratin protofilaments, right, that are bonded together with these disulfide bonds. You reduce them, so now you break all of them. Okay, you actually just break some of them, right? And then you can curl the hair into the shape you want. My little plus thing, that'll go away in a minute. in the shape you want, and then you reoxidize and remake those disulfide bonds so it stays in that shape. Okay? So that's actually how this works. Yes?
Is that how curly hair is naturally?
How, what was that?
Curly hair is naturally.
Curly hair naturally is just going to have, I mean, it's not going to be a reduced and oxidized thing. It's just going to come and you're going to have the disulfide bonds offset slightly. Okay, questions? All right, so that's how a perm works there. You know how Nair works? Nair, right, which is going to dissolve hair away, does the same thing, except that you're doing this reduce step, and it's a stronger reductant. So you do indeed reduce it all like this, okay? You reduce all of it, and the hair falls apart into small little pearl filaments and comes away from each other.
How does it fall out of the but it falls out from your cuticle like that?
I mean, you're going to get some into your hair cuticle? Follicle?
Yeah.
Follicle, yeah. But it's good. It's gonna get. I assume that it gets down into the follicle a little bit, right?
Okay.
I mean, it comes back, right?
Yeah.
Any questions? Okay, just an example of why disulfide bonds are important. I'm going to go on and start talking about protein folding. Okay. So proteins have these shapes. We've talked about these shapes, right? Proteins fold into specific shapes that have function. All right, I've told you before that I like to view proteins as these Pac-Man shaped molecules, or at least a lot of them, like enzymes, where you've got this kind of giant structure that holds a series of functional groups and a small number of functional groups in the right place in the active site. Okay, so it's a structure to do that. This is actually smaller than that, but what you can imagine is that this thing has no function unfolded. And that this down here would have function, whatever that function would be. This is small, so it would be binding something, I guess, folded. All right, and there would be a series of steps this would go through to figure out what the right folded shape is. Okay. So what I want to do, finish up with probably finishing up with this today, is think about what would that be. What would that mechanism be by which this protein would go? Because it gets synthesized in a straight line, right? It's a straight line when it's synthesized, and then it has to find this shape. Now, in reality, it could be being synthesized and it could be folding while it's being synthesized as well. And for some proteins, that certainly happens, that they're synthesized and they're folding at the same time. So let's think about a possible model here. Let's think about a model where a protein samples all possible arrangements, okay? So you imagine that it's going to fold, it's going to get to its lowest energy state, we think is where it's going to fold to. And so maybe it's going to sample all possible things it might do. Now, I want to remind you that we've got these phi and psi angles, right? And the global shape of our protein, where that backbone goes, is determined by what that angle is and what that angle is. All right, so let's think about what might happen for that. So let's pretend that proteins fold by sampling all possible shapes and falling down into the most stable one. So it's going to go ahead and try and find the one that's most stable by trying all of them. How long would that take? Yes, but we're gonna calculate it and find forever. Okay, we're going to define forever. So let's make ourselves some simple assumptions here. let's assume a 100 amino acid protein, which is a pretty small protein, okay? In the computational work I do, we're sampling a series of kind of relevant proteins that we're doing. I have 700 of them that I'm looking at. I don't know that any of them are less than 100 amino acids, okay? If some are, it's less than 10%. All right, so 100 amino acids, pretty small protein. All right, how many angles do you want? How many possible angles do you want to consider here? How many for phi and how many for psi? How many positions would you like to do? You're not answering me because you're shy or because you're not with me.
Like, is there an actual correct answer, or are we just guessing?
Well, you're going to give me an answer. I'm going to tell you it's too large. But go ahead and give me an answer. 3, too much. Let's just do 2. Okay. So let's say there's 2 possibilities here, right? Okay. So it could be either this or this, and this one could also be this or this, whatever this and this is. Right? Clearly, there's a lot more than that, right? It's not 360 degrees because we know some of those angles are going to run the peptide into itself, right? And we even know it's smaller than that because only some of those are going to be pretty good for alpha helices. Only some of them are going to be good for beta sheets, right? So we could limit it. It could make one or the other. But let's assume 2 possible angles for each rotatable bond. All right, so for each amino acid, phi is either this or this, and psi is either this or this. How many bonds do we have then in our 100 amino acid protein? I heard the word 100. I heard the word 100. What number went before that? 200, right. Each amino acid has 2 bonds, and it each has 2 possibilities, right? So that's 200 angles. Right? So we get 200 angles, each of which has 2 possibilities. It could be this or that, right? So if we were thinking about it, we have 2 possibilities for the first angle, right? Times 2 possibilities for the second angle, times 2 possibilities for the third angle, right? 200 times. So that's 200, or that's 2 to the 200, right? Okay, so that's 2 to 200 possibilities for each of those in this super simple system, right? 100 amino acids, a small protein, and we only gave each angle 2 possibilities of what it could do. Let's go ahead and put that number in our calculator. I think I remember that this doesn't cause your calculator to blow up. So 2 to the 200. My calculator didn't blow up, so yours probably didn't either. So I've got 1.6. With all our assumptions, we don't even really need more than one significant figure, times 10 to the 60 possibilities. Okay? Now, when I do computer simulations, we do 1 femtosecond steps. All right, so that's 1 times 10 to the negative 15. Make sure I've got that right. Pico, femto, yeah. What? Okay. And that's really on the time scale of the reason we take 1 femtosecond steps because we're actually on it's on the time scale of the stretching of a bond. All right. So do you think that protein folds on the time scale of the stretching of a bond? Get your bonds doing this. Do you think the whole protein can fold on that time scale? No, it's much, much slower than that. All right. It's going to be at least nanoseconds. Some number of nanoseconds. It's probably microseconds. Okay, not femtoseconds. But let's assume femtoseconds anyway. Okay, we know it's not possible, but let's assume that. So let's assume the protein can sample a conformation, one conformation each femtosecond. So, I don't feel like I'm spelling that right. Femto. Anybody know? There it is. All right, got it. I love spell check. All right, so let's assume that we can each femtosecond. That's way, way too fast. Okay, possibly a million times fast, times too fast, possibly more than that. All right, so how long would it take to sample all these conformations? Yes. In years. All right, so we had 1.6 times 10 to the 60 conformations. And we're going to do one conformation in 1 femtosecond. Okay? And I know that there are 1 times 10 to the 15 femtoseconds in 1 second. Okay? Guys with me? All right, we'll cancel our units, conformations, femtoseconds, seconds. Well, not seconds yet. Take our seconds back. We know there are 60 seconds in 1 minute. We know there are 60 minutes in 1 hour. We know there are 24 hours in 1 day. And we know that there are 365 days in 1 year. We get to divide by a lot of things, right?
Yeah.
You guys okay with my dimensional analysis? Did I make a mistake?
You said 6 minutes. Yeah.
Oh, 60 minutes. Yeah. Let's get all the zeros we can on the bottom here, right? All right, so 1 times 10 to the 60. They're all 1s on the top, aren't they?
Mm-hmm.
All right, divided by 1 to the times 10 to the 15th, divided by 60, divided by 60, divided by 24, divided by 365. I've got something to the 5 to the 37. Is that what other people got? So 5, I'm just going to say 5 because we've got so many estimates here, right? We don't need more than one significant figure. 5 times 10 to the 37th years. How old's the universe? Anybody know this?
More than that.
Well, how long? Anybody know?
13 billion.
13 billion years, right. Universe is 13 times 10 to the billion, 9 years old. Universe is there, okay? So can it work?
No.
Can this be the way proteins do that? No, because it would take way too long, incomprehensibly too long. So proteins have to fold by some sort of smarter method. There has to be some sort of more directed method that causes it to happen. So it would take way too long for proteins to fold if they sampled every conformation. Every conformation and then stuck with the most stable one. All right, so how do proteins fold? we're going to go with two. We're going to go with two kind of hypotheses on this. We're not going to settle them, but we'll talk about two hypotheses. One is that they form into their secondary structures, which is kind of a local structure, right? You got those hydrogen bonds close. They form their secondary structures, and then their secondary structures come together to form the tertiary structure. And then beyond that, once you have protein units, they could come together to make a quaternary structure. We're not going to worry about that. Those two pieces would float together in solution. Another theory out there is that all the hydrophobics get on the inside and the hydrophilics are on the outside. That's called a molten globule. And then they settle down into the final structure. Okay, so all the hydrophobic amino acids get in the middle of the protein surrounded by the hydrophilic residues. Notice that I use residues for amino acids. I didn't really define it. I would also, if I was talking about DNA a lot, I might talk about residues there. Residues are kind of like units, but residue is another way of doing that. So they get surrounded by the hydrophilic residues, and then the protein kind of wiggles around and gets into its final shape. Let's say the backbone. And I'm not really talking about the amino acids might be interacting to get into that final shape, but really you get into that final shape. And if you look at these things in solution, especially in simulation, I expect on the inside of the protein, those side chains are fairly locked in, but on the outside, they're just swinging around all over the place. You'll often see, unless they have something to bind to. All right, and so probably, sorry, a little whiplash there. And the truth is probably some mixture of the two of these. All right, so how many people have, so this is the protein folding problem. How does this work? And the challenge for a long time was that, how can we predict what a protein shape would be from its primary sequence? All right, this is something when I was in grad school, actually, Dr. Sarisky was working on this in her grad work. People have been working on it for a really long time, and up until about 5 years ago, okay, people thought we were still at 40 years off. Okay, and then with machine learning, has anybody heard of AlphaFold? Google Mind, whatever this, the Google research group came up with a program called AlphaFold that actually has largely solved the predictive part of it. All right, so they can take the primary sequence and they can often get the right fold for it, very, very often. All right, but it's pattern matching of this is what's out there. And then I don't know if you know much about this machine learning stuff, but a lot, especially the higher end machine learning, it's like you put a bunch of stuff in, you've trained a model, and stuff comes out, and you don't really know what's going on the inside very well. So it's a predictive model. It's really amazing, right, that we can do this, but we still don't really know how it happens in the cell. Does that make sense? You guys have a good weekend.